Okay, so first of all, Nikos and everybody, thank you very much for giving me this opportunity to present the information, the data about the endothelium, the vascular endothelium, in the immunopathology of airway viral infections and asthma viral exacerbations, which we know that are clinical problems in patients with asthma and other chronic respiratory diseases. And what I may say at the very beginning of my talk is that we all concentrate mostly on epithelium, on bronchial epithelium. And this is obvious because we know that this is a strong source of inflammatory cytokines and all other mediators, which then thereafter shape the immune response in many circumstances. And we know, of course, that bronchial epithelium is the first target for viral infections, respiratory viral infections. But we actually are not allowed to forget that in the pulmonary tissue, taken all together, we have many, many vessels which are covered, which inner side is covered by endothelial cells, by vascular endothelium, which is a very, very strong and potent source also of immunity. And what I'm going to show you for the next several of minutes, it may be somehow engaged in viral respiratory infections as well. So the title of my talk is the viral infections and the endothelium, vascular endothelium. And today I'm going to focus especially on rhinoviral infections. Okay. So just to have a view, in our body, we have very complicated circulatory system, which vessels are covered by the endothelial monolayer, the monolayer which is made of endothelial cells. And each inner side of our vessels, nevertheless, this is the vein or this is the artery, it is covered always with integrated endothelial monolayer, which is quite nicely visible here on this picture. This is also, if we want to compare it to the bronchial epithelium, I will refer to bronchial epithelium because we all are concentrated on it as well, and this is very, very familiar to us. This is also a kind of barrier because we perceive bronchial epithelium as a kind of barrier. And endothelium, this is also the barrier between the tissues and between the blood and everything which is flowing with our blood. Endothelial, vascular endothelium has got a lot of different functions, like white cells trafficking, like vascular permeability, like vascular tone. The endothelium may regulate vascular tone. It may regulate angiogenesis. It also takes a really huge part in inflammation. It has got the metabolic functions. It is also associated with smooth muscle cells proliferation, with fibrinolysis, of course, with platelet activation, and with thrombosis. In the physiological situation, endothelial cells are constantly working, but they are not inflammatory activated. When we have the non-inflamed condition, it looks like that. But when we have the inflammation in the tissue, endothelium serves as a barrier between the tissue and the blood, and of course must be inflammatory activated to let all cells coming from the bone marrow through the bloodstream to the inflamed tissue to give the help to the danger which may occur in our tissues. So immunopathology of endothelium looks like that in chronic inflammatory condition, vascular endothelium may be involved in immune cell trafficking, antigen presentation, it has been shown, inflammatory responses and vasculitis, angiogenesis, formation of secondary and tertiary lymphoid organs, which seems to play a very, very important role in the local homeostasis, immune homeostasis within the tissues and within vessels. And also lately it has been, we are focusing on the endothelial to mesenchymal transition. So endothelial cells in chronic inflammatory conditions may change their origin and go, for example, into cartilage formation, endochondrial ossification, or change into the smooth muscle cells. And of course, as I mentioned, endothelium looks to be a very, very potent source of inflammatory mediators. And here on the right-hand picture, you may see the release of inflammatory mediators, inflammatory cytokines in resting lung endothelium. And you can see how much of IL-8, for example, which is a strong chemoattractant, yes, MCP for monocytes, MIP, or IL-6 is being produced only in resting endothelium. So this is a strong and potent source of inflammatory cytokines. So the second introduction is encompasses the viral infections, respiratory viral infections, which we actually are exposed to, especially during autumn or the winter time. We all are exposed to many viruses which are flying in our air and then gets into our airways and induces inflammation. And usually we meet rhinovirus and coronavirus. And now we are knowing that, yes, the coronavirus SARS-CoV-2 is very dangerous. And this is a kind of exceptional so far. But we have been, during our lifespan, we have been exposed to many low pathogenic coronaviruses, like, for example, to 9E. Viruses, respiratory viruses like rhinoviruses or coronaviruses, they are responsible for many episodes of common colds. But in certain people, like, for example, having asthma, they may be much more harmful. And we know the problem about the rhinoviral exacerbation of asthma, which may be very dangerous for especially uncontrolled patients. And we know that even 60 to 80% of viral exacerbation of asthma is made by rhinovirus, but only 4 to 8% asthma exacerbations is evoked by coronaviruses. So, of course, we know that the bronchial epithelium is the main target for all of these respiratory viruses, because bronchial epithelium is equipped with many receptors for these viruses. And we know that infected epithelial cells also release a lot of different inflammatory cytokines to the bronchial mucosa, and then shape the immune response and inflammatory response. But we have to be aware that in the bronchial mucosa we have a lot of vessels which are perforating the bronchial mucosa. We have to be very aware that in alveoli we have a very strong closeness between epithelial cells covering the alveoli and endothelial cells of vessels through which the exchange of gases, ions, and especially gases like oxygen or carbon dioxide occurs. So we can see the very close proximity in the localization between epithelial cells and endothelial cells here, and epithelium here, and endothelial cells in vessel which perforate bronchial mucosa. And we have to be aware, speaking about the rhinovirus, that not only epithelial cells have the expression of ICAM1, which is the main receptor for human rhinovirus, but also endothelial cells are very, very largely equipped with this ICAM1 molecule. Of course, from the physiological point of view, this is very, very important because ICAM1 is engaged in cell trafficking and cell recruitment from the bloodstream to the tissues. So endothelial cells must be actually equipped with this ICAM1 molecule. But from the viral point of view, this is an entry receptor for rhinovirus. So having in mind all this information, we concentrate our minds on the possibility of the infection of vascular endothelial cells, not only epithelial cells, but lung vascular endothelial cells by rhinoviruses. And we asked the second question, we asked the question whether human rhinoviruses may infect lung vascular endothelium and what might be the consequences of endothelial infection. So this is a good approach which may define the endothelial cells in chronic inflammatory conditions. This is a very good paper showing this, and they paid attention on the pathological angiogenesis, which may be related to vascular endothelium engagement in the chronic inflammation, immune cell recruitment, MHC2 class expression, immunoregulatory properties, endothelial to mesenchymal transition. But in our approach, we concentrated mostly on the potential consequences which may be related to the viral infection of our endothelial cells. So this is a kind of plan of my talk today, and I'm going to follow all these, let's say, things which we want to describe the endothelial cells with in the viral infections. So first of all, we have to perceive endothelial cells as a target also for respiratory viruses, because endothelial cells have been shown to express ICAM1, as I said, which is the receptor for rhinovirus, but also aminopeptidase N, which is a receptor for coronavirus 229E, which is this low pathogenic coronavirus. But it also has the expression of the dipeptidyl peptidase 4, which is a receptor for SARS-CoV-1. And we also, what I'm going to show later, endothelial cells may, in some circumstances, express ACE2 receptor, which we all now know because of the coronavirus SARS-CoV-2 pandemics. So the first very basic question which we asked was, may rhinovirus infect lung vascular endothelium? And we developed a very easy model in which we incubated our endothelial cells with human rhinovirus 16 for 3 hours. Then we did the washout, and then we further cultured cells for 5 hours, 24 and 72 hours. So this is our basic model, which is going to be repeated and repeated all the time. First, this is our first data, which were published in APMIS and then in Allergy. So the first publication concentrated mostly on umbilical vein endothelial cells, but then because we want to concentrate mostly on the immunopathology of respiratory infections, our research concentrated then on the microvascular lung endothelial cells from human. And in the very first data, you can very nicely see that human rhinovirus 16 may infect endothelial cells, which may be related to the MOI of the virus. And then during the time, we can see the increase of the number of viral particles, but actually it peaks about 10 hours and then it drops out. And this is a very nice difference between MOI3 and MOI1, as we can see in umbilical endothelial vein cells. And we can see that if we block ICAM1 with antibodies, the uptake of the virus was very, very reduced. The same we may observe in human microvascular lung endothelial cells, and the dynamic, interestingly, looks the same, but the peak value is being established in the 5th hour as compared to HUVEC, which was in the 10th hour actually. And then if you block ICAM1 with antibodies, you may also inhibit the uptake and the replication of the virus. We also shown that for the very short time, ICAM1 surface expression is decreased, probably because of the kind of occupation by the virus. And because we all know that the optimal temperature for rhinovirus replication in epithelial cells is 33 to 35 degrees of Celsius, we tried to check this in endothelial cells, but unfortunately, because of some reason, we could see the capture of the virus, but we could not see any increase of the viral particles. So speaking about endothelial cells, this temperature is too low. Viral particles, they have RNA, which is a strong ligand for toll-like receptors and Rig-like receptors, which we know that all belongs to the PRRs. We could nicely see that rhinoviral infection of endothelium increased the expression of TLR3, Rig-I, and MDA5, and you could see that the dynamics was really repeatable here. So somehow we achieved the peak of the expression, and then we can see the drop in the with the lowest value in the 72 hour. And of course, if we block ICAM1 with antibodies, we also could see the inhibition of TLR3, Rig-I, and MDA5. So it means that RNA of virus do not enter the cytoplasm. The same we observed in lung endothelium, and we nicely see TLR3, Rig-I, MDA5, but also TLR7 and NOD2 mRNA expression, and the expression is quite nice here, but actually we could not see any expression of TLR8 receptor. So let's stay for a while in the toll-like receptors. We know that TLR3 and 7 and 8, these are endosomal receptors for RNA. So each foreign RNA which gets to the site, which enters the cytoplasm actually, or is encapsulated here, it may be recognized by innate immune receptors. And then it has a really great consequences, because it starts all signal cascade, which at the end of the day leads to the production of interferons, which are antiviral, of course, agents, and also it leads to the inflammatory activation. So such cell which pattern recognition receptors are induced by their ligands, each cell will produce interferons and inflammatory cytokines. And indeed, it occurs when the virus, the rhinovirus, enters the cytoplasm. It is encapsulated by the endosome, and then when the RNA is released, it may affect TLR3, TLR7, and 8, and then lead to the inflammatory activation of the cell. And the second, let's say, part of this antiviral defense, because this is a kind of antiviral defense system, in the cytoplasm, when the viral particles are released from the endosomal compartment to the cytoplasm itself, there are also the complex of receptors which may catch the RNA of the virus and may be activated and may activate again inflammatory response and interferon production, and additionally it may also induce apoptosis by mitochondrial way of apoptosis. And let's have a look at Picornaviridae. This is a family to which the rhinovirus belongs. It mostly affects MDA5 receptors. So we mentioned about the entry receptors. So endothelial cells may uptake the rhinovirus. Because the endothelial cells, and this is very nice because we have to also change the understanding of endothelium. This is not only the barrier, this is not only the source of inflammatory cytokines, but this is a kind of very complex mechanism and a kind of sensor of pumps due to the possessing of pattern recognition receptor complex. So endothelial cells may really nicely sense the viral particles, and then it may have a really huge consequences in its activation. So let's concentrate on the sensor of pumps and antiviral. It may lead to the have a look on the dynamics, because the dynamics of the interferon production in endothelial cells upon rhinoviral infection looks differs a bit as compared. So this is very, very important for the antiviral defense of cell before the adaptive immunity will be turned on, because we know that it takes a while to produce antibodies, yes? It takes a while also, maybe not so very long while, to recruit all NK cells or other cells which may fight with the virus. So cell itself has to be a kind of prepared for the fight with the virus itself. So interferons which may be produced by rhinoviral infected endothelium, they are very, very important, because they, first of all, can act on the healthy cells and prepare them to obtain a kind of resistance to the viral infections. And also we observe the autoregulation, that interferon may autoregulate the expression of antiviral agents in the cell which is already infected. So we can see that interferon, it may increase the expression of protein kinase A, which then at the end of the day will inhibit the translation of mRNA and production of new viral particles. Interferon may also induce oligoadenylate synthase, OAS in short term. And this is very, very important, because this enzyme may degrade the already produced and synthesized RNA chain. So the newly produced RNA of the virus may be cut and divided at the very, very beginning. So it is not being able then to be used for the whole viral particle production. So actually we checked whether this appears in our endothelial cells. And first of all, we checked whether endothelial cells may express both of these agents. And indeed, it may express, you can see the expression of OAS and PKR in the confocal microscope. So what we observed is that during the infection of endothelial cells with human rhinovirus, we could see the same dynamics of the increase of OAS1 expression mRNA, which peaks actually altogether with the peak number of viral particles inside of the cell, and then it drops down in the function of time. You can see very nicely that this increase of OAS protein inside of the cell is visible in the, is marked actually in the 24 and to 74 hours after the infection. It seems to be also a bit activated at the very beginning, but the difference is not so huge. And here you also can see that in the HRV infected endothelial cells, the expression of OAS is much higher as compared to MOC cells. And the same actually is with our kinase, protein kinase R. The dynamic is the same, but the, let's say, fold of increase is lower, which is also marked here, but still we can see the increased expression of PKR, both in the flow cytometry and in the confocal microscope. So this is very, very important because we can see that a viral infection, rhinoviral infection in endothelial cells may activate cellular antiviral defense mechanisms based on OAS and PKR expression. So maybe, I mean, this is our hypothesis that this dynamics that you can see, the increase of viral particles and then sudden decrease, it may be a kind of associated with the activation, with the successful activation and effective acting of these intracellular interferon-based mechanisms of antiviral defense. Okay, so the next approach, next feature of our endothelial, vascular endothelial cells, which may have a very, very huge importance in viral infections of the respiratory system. This is this has got as inflammatory and immunoregulatory activity. And at the very beginning, we showed that infection with human rhinovirus of vascular endothelium, it may increase the expression of RANTES, and IP10, which then is accompanied by the production of the protein, dependingly on the dose of the virus. When you block ICAM1, still you can almost 100% inhibit the production of these chemokines and inflammatory agents. The same we observed in human lung microvascular endothelial cells. And just to remember, this is a basic production of inflammatory cytokines in resting endothelium. So we asked the question, what's going on with other inflammatory cytokines? So this was the work actually of Adrian Gajewski, which is a postdoc in our lab. And he did some bioplex assessments, measurements, and we concentrated on 2070 cytokines. And here he also developed a kind of system of the analysis of the fold increase of cytokine production and the intensity. So how much of the cytokine is produced? Because we know that IL-6, IL-8, they are produced in very, very large amounts. Contrary to IL-4, for example, or IL-5, which are immunoregulatory cytokines, which usually are produced in smaller amounts, but actually it doesn't bother them because they really act in the immune synapse. So locally, they may have much larger concentrations. So here you can see the basic production of the proteins, inflammatory cytokines. And here you can see what's going on with the inflammatory activity of endothelial cells, which are infected with human rhinovirus. So IL-6, which is actually expectable. Now IL-6 is being used as a kind of marker of the heaviness of the COVID-19 disease. Yes, based on this we may, and this is also based on this we also base our treatment. Actually, we can see the growth factors which are being really expressed. Of course, interferon, which is quite nice because we need interferons when the cell wants to fight the virus. We can also see the very huge increase of chemokines. So the inflamed tissue needs the recruitment of the inflammatory cells to fight with the virus. But also we may also see actually nice increase of immunoregulatory cytokines like IL-4, IL-5, IL-12, IL-17, interferon gamma, but not IL-13 and IL-10. So Adrian showed that human rhinovirus may very strongly activate inflammatory phase of our vascular endothelium. And if you can imagine, on one hand, we have the epithelial cells, which when are infected, they release all this stuff into the bronchial mucosa. But on the other hand, when probably endothelial cells are infected also with the rhinovirus, which may be possible, which I'm going to show you in a few seconds, then we have another maybe comparable strong source of inflammatory mediators coming up from the vessels within the bronchial mucosa or within the lung alveoli. Antigen presentation, we actually just about to start this this fragment of our research. So I'm just going to show you very first, let's say, data that human rhinovirus may seems to be able to enable the endothelial cells to present the antigen. Of course, we know that resting endothelium does not, as you can see here, express MHC class 2, so it cannot be antigen presenting cell, but in some circumstances like interferon gamma, for example, action or other inflammatory condition, we know that some not professional antigen presenting cells, they may gain the ability to present the antigen. So it also looks that rhinovirus may increase MHC class 2 mRNA expression and CD53. I'm going to tell you why this is so important. So first of all, maybe rhinovirus may make endothelial cells to gain the ability to present the antigen, which is having very serious consequences, because then it would indicate that endothelial cells may directly affect the response, the specific response, adaptive responses. And CD43, why this is important? Because CD43 is the costimulatory molecule which is being used in the contact between memory T cells and endothelial cells, for example. Yes, so CD53, when it is expressed, it does not affect naive T cells, but it may affect and present the antigen to the memory cells. So it is it might be very important booster of adaptive immunity development during the viral infection, and this may be the consequence of this endothelial future. Okay, so having described all these abilities of the endothelial cells to be infected, to be inflammatory activated, to induce the antiviral mechanisms, we have to ask the question whether there is a possibility of any transmission of the virus from the epithelium to the vascular endothelium. Because we know that the main target for the rhinoviruses is epithelial, bronchial epithelium. So at least at the very beginning, we tried to set it in the laboratory. And Robert Szewczyk, a PhD student, this is a very small part of his large research, especially on the coronavirus. But this I'm not having the time to present it today. So he proved that when the healthy endothelial cells were exposed to the supernatants taken from the infected epithelial cells, it may really infect our endothelium. And this you can see the number of the viral particles when endothelial cells were exposed to the 5-hour supernatant after the infection of epithelium. And here you can see that the number of viral particles in the endothelium is much higher when the endothelium is treated with the epithelial-derived supernatants after 24 hours after the infection, which is quite logical. And this is also associated with RANTES production and IP10 production. So here we could have a kind of proof that at least in vitro condition, the transmission of the rhinoviral infection from the bronchial epithelium to the vascular lung endothelium may eventually occur. Okay, so the next very important field of our research concerns the endothelium as a barrier. We also are very concentrated on the epithelium, bronchial epithelium, which definitely is a barrier which divides our inner body from the outside world. And this barrier has to be very, very efficient. But of course, endothelial cells, this is also a kind of barrier, actually much more vulnerable than epithelial cells, but still this is a barrier. And if this barrier does not work, you may have a very, very huge consequences. So we also, this is another approach of our understanding of the endothelial cells, that this is a kind of self-renewing multi-generational barrier cell system. And this is what we're going to concentrate for the next few minutes. So we can see here that this is a kind of reflection of the endothelium, which covers the inner side of the vessel. And of course, the endothelium, similarly to epithelium, has got the expression of the same proteins, but in difference, the protein, which is mostly important for the barrier functions of endothelium, is at VE cadherin and adherent junctions. But tight junctions are also present in vascular endothelium, and especially they have a very huge and important action in the blood barrier endothelium. But we are in the lungs, so we're going to concentrate on lungs. And actually, we proved several years ago that the endothelial barrier may also be, similarly to epithelial barrier, regulated by inflammatory cytokines. And here you can see very nice view that IL-33 was able to partially and transiently decrease the endothelial barrier, and then it went back again to the same status. Contrary to IL-4, which we know that both they are allergic cytokines, but IL-4 changed the barrier functions, decreased the barrier functions, not reversibly. So Mateusz Gawrysiak, he is the PhD student as well, which touched this topic. And he was especially interested in the influence of human rhinovirus on the barrier functions and regeneration of human lung vascular endothelium. So he used the same actually model, and what we found, and this was published in Allergy last year, that he found that rhinovirus may nicely decrease the integrity of the endothelial monolayer, which is accompanied with the increase of the permeability, which is also accompanied by the decrease of VE cadherin expression in the confocal microscope. This is 5th hour, this is 24th hour, this is MOC cells, and these are cells which are exposed to rhinovirus, which you can see that the barrier is really, really lost, and then that it is that the VE cadherin expression is highly decreased. Of course, rhinovirus may kill the cells, and here you can see that rhinoviral infection of the endothelial cells led to the increase of apoptosis. And the second part of Mateusz' paper is that he concentrated on the regenerative and migratory capacities of lung endothelial cells infected by human rhinovirus. And you can see here that when the cells were treated with the rhinovirus, the wound healing was delayed as compared to the control cells or as compared to the intact virus. And here you can see also that this wound healing delay was associated with the inhibition of migratory capacities of the cells. But so we may say that the rhinovirus may destroy the endothelial barrier, and this is truth, because we could see all consequences of the impairment of the barrier, like edema, for example, of the bronchial mucosa or nasal mucosa. We all have discharge and stuff. But we also ask the question, what's going on later on? So what are the long-term effects on the angiogenesis and endothelial cells infected by rhinovirus? And this is a kind of reflection which we have because we know that the asthma is associated with remodeling and with increased angiogenesis. And it is tried to be associated also with repeatable rhinoviral infections. So we asked the question, what's going on with the integrity of the endothelial monolayer after the virus is a kind of eradicated, because we can see here that this virus number, particle numbers is decreased. So it may a kind of suggest that the system itself may a kind of eradicate the virus. And what we observed that actually at the very beginning, the monolayer integrity is very, very nicely, let's say, destroyed here. But we can see that thereafter, during the next few days, the same model kind of restores. Then actually in 6th day, we can see quite nice and the same monolayer. So this is a generation of cells which have been already infected, but then somehow this model could fight with the virus. And here you can see the reflection of integrity of this monolayer. Here you can see what's going on with the mock cells, and here the first decrease, and then even the integrity of infected cells and monolayer is increased. And this is what actually Nikos kind of showed several years ago, that when he did the experiments using epithelial-derived supernatants and induced endothelial cells, and he showed that these supernatants may induce, for example, the production of VEGF. Actually, what we do here is we check the direct effect of rhinoviral infection. And here you can see that this restoration, which is seen here on this picture here, may be associated, and this is our hypothesis, which we're going to publish very, very soon, that it may be associated that after 2 or 3 days, we could see the increase of mRNA expression of VE-gatarin and Claudin-5. We could see the nicely increased expression of angiogenic factors, and we could see the increased expression of receptors for these angiogenic factors. So it seems that human rhinovirus infected endothelial cells, in some point, they may be open for the new angiogenesis and for the restoration, because they themselves produce angiogenic factors, and they are open for these factors, that they can increase the expression of receptors for these factors. Here you can see that the protein expression is also seen. Maybe we can see the higher proliferation in 6 hours, in 6 days after the human viral infections, as compared to the mock cells. So summary one is that we showed that rhinovirus may infect the vascular endothelium. It may induce interferon, chemokines, growth factors, inflammatory and modulatory cytokines. It may induce and activate all pattern recognition receptor apparatus. It may impair endothelial barrier in the short time, but maybe it kind of restores this barrier later on. And it impairs short-term endothelial regeneration and migration. And the last, actually, but not least, is the field of the susceptibility of endothelium to the cytokine modulation, because we know that during, for example, asthma or viral infection, endothelium may be exposed to the very, very large amounts of cytokines, which are produced by epithelia, by other immune cells. So it may also be exposed. And so coming back to actually our field of interest, to the asthma, we know that IL-33 is a cytokine which plays a really, really important role in asthma. So that's why we concentrated. And the other point is, I'm going to check the hour. And the other point is that asthma may be exacerbated by rhinoviruses mostly, and in very, very low extent by coronavirus. And the question is actually, why is it so? Because we are also exposed in our environmental conditions on coronaviruses. So why rhinovirus may be so harmful for asthmatic patients and coronavirus not? Actually, we know that even though the patients with asthma have got chronic respiratory diseases, they seem not to be in the higher risk of COVID-19. development, as compared to COPD patients, for example, which also are having the chronic inflammatory condition in the respiratory system. So we had a lot of very, very exciting and interesting questions. And we wanted to concentrate on in vitro models. What might be the effect of IL-33 on the rhinovirus infection? So we asked the question. The first question was whether IL-33, what it may do to the human rhinoviral infections of endothelium, and how it may affect coronavirus receptors. So this was published this is the work by Adrian Gajewski again, published in Allergy, and he proved that IL-33 may increase ICAM1 expression. And this is obvious, but actually if we consider that ICAM1 is a receptor for rhinovirus, then we may imagine what might be the consequences. If we have more IL-33, if we have more ICAM1 expression on the endothelial cell in asthmatic patient which has got higher numbers of IL-33 in the respiratory system, then we may assume that the capture of the virus may be much more actually affected. And we can see that actually when the endothelial cells were pre-treated with IL-33, indeed the number of viral particles inside of the cell which were much, much higher as compared to rhinovirus only. So we proved that IL-33 may increase the capture of the virus. So at the same time, more viral particles gets into the cell. So we also could see the reflection of it in the production of cytokines. You can see that IL-6 was even more produced in the environment of IL-33. The same with growth factors. The same with IL-8, which we actually IL-8. As well as other cytokines. We can see that also IL-12, IL-17, and IL-3, IL-4, IL-5, they were also upregulated. But this is very, very interesting that interferons were not upregulated. Yes, that IL-33 increased the inflammatory cytokine production, but did not affect interferon production. And we know that interferons are needed for the self-defense against viruses. So we can't see that IL-33 increased neither interferon type 1 and interferon gamma, which may have really huge consequences. And coming to the last part of my talk is that we wanted also to check whether rhinovirus and IL-33 may somehow affect coronaviral receptor expression. And we showed that endothelial, lung vascular endothelial cells may actually express the receptors for all main coronaviruses, including coronavirus SARS-CoV-2. And another puzzle to the story is that we can see that in the environment, we have a period of time during the year when at the same time we have rhinoviruses and coronaviruses in our environment, and we may be exposed to both types of viruses. So the question is whether we are, as our organism, is a kind of field of the environmental interplay between these viruses in the real life. So Isabella Gulbas, which is also doing a PhD in our laboratory, took over this topic. And she asked this question. So, and this is the paper which is being revised in Allergy at this moment, and we have the first revision. So, just briefly, Isa showed that rhinovirus, first of all, rhinovirus may increase aminopeptidase receptor expression, which is for coronavirus 229E. So this is very nicely seen in confocal microscope as well as in the facts here. But let's have a look what's going on with IL-33. When cells were pre-incubated with IL-33 and then human rhinovirus was added, so we could see that this increase was actually inhibited here. Yes, so IL-33 may inhibit the expression of a coronavirus receptor induced by the human rhinovirus. Okay, so this is what we can see here. What about MERS receptor for MERS, dipeptidylopeptidase 8? We can observe the same dynamics. So this is mRNA expression. A rhinovirus may increase it. It may increase the protein expression in FACS and confocal here. But when we add the IL-33, this increase is inhibited, which is very nicely visible here. What about ACE2? So we can see that rhinovirus may increase the expression of ACE2 for coronavirus SARS-CoV-2, but the dynamics is quite different. This is very rapid dynamics. This is like an interferon-inducing dynamic. And in the 24 hours, IL-33 very strongly inhibits the increase of the expression of ACE2 receptor for SARS-CoV-2 virus. So we can see that the virus may increase it. The expression is quite different as compared to the other receptors. And then IL-33 may inhibit this actually increase, which is quite nicely seen on this FACS picture. And here you can see the three-dimensional, but the expression of this receptor, but we actually don't have too much time. And then what about TMPRSS2, which is a kind of co-receptor, very, very important for cleavage of SARS-CoV-2 virus. The cells do not have resting expression, but when you add human rhinovirus, the expression very, very nicely occurs here. So we can see the expression here, and IL-33 decreases the expression. This is very, very nice data. So we also wanted to see what the IL-33 does with other receptors. And speaking about ICAM1, we could see that rhinovirus really increases this expression very nicely, which is already known. But fortunately, IL-33 is able to decrease it. So we can see the divergence effect of IL-33. Coronaviral receptors are inhibited, but ICAM for rhinovirus is actually increased. And when you block the ICAM1 expression, you also can block these effects. So the last summary is that we found that IL-33, which, let me remind you, is a very, very important cytokine of asthmatic inflammation, it may enhance the in vitro infection with human rhinovirus. It potentiates chemokine, growth factors, inflammatory and modulatory cytokine release. It does not, unfortunately, it does not affect interferon-dependent mechanism, and it increases both resting and human rhinovirus-infected ICAM1 expression. So overall, maybe, this is our hypothesis, this is the reason of heavier rhinoviral asthma exacerbations, that the presence of IL-33 on one hand may, let's say, initiate very strong inflammatory response, may induce the enhanced capturing of the virus, but does not induce stronger antiviral response. But on the other hand, what is very interesting, IL-33 decreases receptors for coronaviruses. Maybe this is a reason that in general we observe much less coronaviral asthma exacerbations in, let's say, normal world. And maybe this is also the reason that we can observe less risk of the COVID-19 infections in our patients. But this is of course, the very, very loose hypothesis which we are going to, let's say, check in the future. So coming to the very end, the conclusion is that the vascular endothelium of lung vessels has a potential to sense the respiratory viruses and potentially orchestrate inflammation during airway viral infections. So not only epithelium, but also endothelium should be considered in this very, very complex, let's say, system. So we should also, this is my final remark, perceive endothelial cells as a quite important part of our innate immunity, because it has got the pattern recognition receptors. It may sense a lot of viral particles and probably other pathogen particles. So it may be really actively involved in the inflammatory response against respiratory viruses. So thank you very much for the very long talk and for your attention. I hope I was brief as much as I could do. And this is my team. So I'd like to thank Izabela Gulbas, Robert Szewczyk, Ola Likońska, Mateusz Gawrysiak, Adrian Gajewski, of course, Professor Kowalski, which has got a huge impact on our research. And thank you, all of you, for your attention.